D. Gene concept. Although the role of hereditary units factors in transfer of genetic characters over several generations in organisms was advocated by Gregor John Mendel, yet the mystery of the hereditary units was unraveled during early 1900s. In 1909, W. Johansen coined the term gene that acts as hereditary units. However, early work done by several workers proposes various hypotheses to explain the exact nature of genes. In 1906, W. Bateson and R. C. Punnett reported the first case of linkage in sweet pea and proposed the presence or absence theory. According to them, the dominant character has a determiner, and the recessive character lacks determiner. In 1926, T. H. Morgan discarded all the previous existing theories and put forth the particulate gene theory. He thought that genes are arranged in a linear order on the chromosome and look like beads on a string. In 1928, Belling proposed that the chromosome that appeared as granules would be the gene. This theory of gene was well accepted by the cytologists. In 1933, Morgan was awarded Nobel Prize for advocating the theory of genes. After the discovery of DNA as carrier of genetic informations, the Morgan's theory was discarded. Therefore, it is necessary to understand both the classical and modern concepts of gene. According to the classical concepts, a gene is a unit of 1. Physiological functions, 2. Transmission or segregation of characters, and 3. Mutation. In 1969, Shapiro and co-workers published the first picture of isolated genes. They purified the lac operon of DNA and took photographs through electron microscope. In 1908, the British physician Sir E. R. Garrett first proposed one gene, one product hypothesis. In 1941, G. W. Beadle and E. L. Tatum, working at St. Stanford University, clearly demonstrated one gene, one enzyme hypothesis, based on experiments on Neurospora crassa. They made it clear that genes are the functional units and transmitted to progenies over generations, also they undergo mutations. They treated N. crassa with X-rays and selected for X-ray-induced mutations that would have been lethal. Their selection would have been possible when N. crassa was allowed to grow on nutrient medium containing vitamin B6. This explains that X-rays mutated vitamin B6 synthesizing genes. They concluded that a gene codes for the synthesis of one enzyme. In 1958, Beadle and Tatum with Liederberg received a Nobel Prize for their contribution to physiological genetics. Units of a gene. After much extensive work done by the molecular biologists, the nature of gene became clear. A gene can be defined as a polynucleotide chain that consists of segments each controlling a particular trait. Now, genes are considered as a unit of function, cistron, a unit of recombination, Recon, and a unit of mutation, Mutan. 1. Cistron. One gene, one enzyme hypothesis of Beadle and Tatum was redefined by several workers in coming years. A single mRNA is transcribed by a single gene. Therefore, one gene, one mRNA hypothesis was put forth. Exceptionally, a single mRNA is also transcribed by more than one gene and it is said to be polycistronic. Therefore, the concept has been given as one gene, one protein hypothesis. The proteins are the polypeptide chain of amino acids translated by mRNA. Therefore, it has been correctly used as one gene, one polypeptide hypothesis. Moreover, genes are present within the chromosome and their cis trans effect governs the chromosome function. Therefore, S. Benzier termed the functional gene as cistron, figure 2.6a. Crossing over within the functional genes or cistron is possible. The cis and trans arrangement of alleles may be written as below. Cis wild. Trans mutant. 2. Recon. Earlier, it was thought that crossing over occurs between two genes. In 1962, S. Benzier demonstrated that the crossing over or recombination occurs within a functional gene or cistron. In a cistron, the recombinational units may be more than one. Thus, the smallest unit capable of undergoing recombination is known as Recon, figure 2.6b. S. Benzier, 1955, found that the cultures of T4 bacteriophage formed plaques on agar plates of Escherichia coli. Normal T4 formed small plaques of smooth edges, whereas the mutant T4 phage formed the larger plaques of rough edges. The DNA molecule of T4 phage consists of several genes, one of which is called R2 region. Formation of rough-edged plaques was governed by two adjacent genes, cistrons R2a and R2b, in mutant bacteriophage. Both the regions function independently and consist of 2, 5, and 1, 5 nucleotides, respectively. In R2A gene, over 500 mutational sites are present where crossing over may occur. Through crossing over, exchange of two segments of DNA occurs. If crossing over takes place within the gene, by mating to R2 mutant of T4 phage, a normal wild-type phage can be produced. Thus, the work of Benzier lends support that crossing over within a gene occurs, which explains that the recombinational unit, Recon, is much less smaller than the functional unit, i.e. Astron. 3. Mutan. S. Benzier, 1962, coined the term mutant to denote the smallest unit of chromosome that undergoes mutational changes. Hence, mutant may be defined as the smallest unit of DNA which may be changed in the nucleotide. Thus, changes at nucleotide level are possible. Figure 2.6c. The smallest unit of mutant is the nucleotide. Therefore, Astron is largest unit in size, followed by Recon and Mutan. This can be explained that a gene consists of several Astron, a Astron contains many Recon, and a Recon a number of mutants. However, if the size of a Recon is equal to Mutan, there would be no possibility in Recon for consisting of several mutants. F-split genes, or introns. During 1970, in some mammalian viruses, e.g. adenoviruses, it was found that the DNA sequences coding for a polypeptide were not present continuously but were split into several pieces. Therefore, these genes were variously named as split genes or introns, interrupted genes or intervening sequences, inserts, and junk DNA. For the discovery of split genes in adenoviruses and higher organisms, Richards J. Roberts and Philip Sharp were awarded Nobel Prize in 1993. As shown in Fig 2.7, a DNA sequence codes for mRNA, but the complete corresponding sequence of DNA is not found in mRNA. Certain sequences of DNA are missing in mRNA. The sequences present in DNA but missing in mRNA are called intervening sequences or introns, and the sequences of DNA found in RNA are known as exons. The exons code for mRNA. After transcription, a limited RNA transcript has the intron sequences present in the interrupted genes. Genes coding for rRNA and trna may also be intervened. The introns are also found in some of the prokaryotes such as cyanobacteria and archaeobacteria, archaea. For some time it was not certain how mRNA is synthesized from a DNA containing introns. Some possible explanations for the mechanism of mRNA synthesis were given. 1. DNA rearrangement occurs during transcription with the removal of introns. 2. During transcription, RNA polymerase skips the introns and transcribes only exons. 3. Individual exon transcribes separately and rejoins to form the complete mRNA. And 4. RNA polymerase may synthesize both introns and exons, and processing of transcripts occurs later on. The transcripts corresponding to introns are removed. Later on, it was shown that the fourth mechanism operates in transcription of mRNA. 1. RNA splicing. Initially, the RNA transcript introns are synthesized, which are later removed by a process called RNA splicing. Figure 2.7. The junctions of intron exon have a GU sequences at the intron's 5' end, and an AG sequence at its 3' O end. These two sequences are recognized by the special RNA molecules known as small nuclear RNA, snRNA, or SNPs. These together with proteins form small nuclear ribonucleoprotein particles called snRNPs. Some of the snRNPs recognize the splice junctions and splice introns accurately. For example, the UI snRNP recognizes the 5' splicing junction, and the U5 snRNP recognizes the 3' splicing junction. Consequently, pre-mRNA is spliced in a large complex called a spliceosome. The spliceosome consists of pre-mRNA, 5 types of snRNPs and non-snRNP splicing factors. Robert and Sharp, the Nobel Prize winners in 1993, independently hybridized the mRNA of adenovirus with their progeny or DNA segments of virus. The mRNAs hybridized the ssDNA of virus where the complementary sequences were present. The mRNA DNA complexes were observed under electron microscope to confirm which part of viral genome had produced the mRNA strand. It was found that mRNA did not hybridize DNA linearly but showed a discontinuous complexes pattern. Huge loops of unpaired DNA between the hybridized complexes clearly revealed the large chunk of DNA strand that carried no genetic information and did not take part in protein synthesis. The adenovirus mRNA contained 4 different regions of the DNA. The B globin genes of mice and rabbits, and trna genes of yeast tyrosine trna consist of 8 genes, 3 of which have been studied in detail. Each gene contains 14 bases, AVTACA, as intron in the middle. In the same way, the pre-runner genes contain introns of 18 to 19 bases. In all the genes, introns are present near anticodon. Similarly, a few rRNA genes are also known to contain introns, and some of pre-rRNA are self-splicing. 2. Ribozymes. For the first time, Thomas Cech in 1986 discovered that prerna isolated from a ciliated protozoa, Tetrahymena thermophila, is self-splicing. Thereafter, S. Altman showed that ribonuclease cleaves a fragment of pre-runner from one end, and also contains a piece of RNA. This RNA fragment catalyzes the splicing reaction, i.e. acts as enzyme. Therefore, this RNA segment catalyzing the splicing reaction is called ribozyme. For this discovery, Cech and Altman were awarded the Nobel Prize in 1989 in chemistry. The best studied ribozyme activity is the self-splicing of RNA. This process is widespread and occurs in T. thermophila pre-runner, mitochondrial rRNA and mRNA of yeast and other fungi, chloroplast runner, rRNA and mRNA of bacteriophage. The rRNA intron of T. thermophila is 413 nucleotide long. The self-splicing reaction needs guanosine and is accomplished in 3 steps. 1. The 3G attacks the 5' group of introns and cleaves the phosphodiester bond. 2. The new 3O group on the left exon attacks the 5-phosphate on right exon. Consequently, 2 exons join and remove the intron, and 3. The 3O of intron attacks the phosphate bond of nucleotide 15 residues from its end, releasing the terminal fragment and cyclizing the intron. 3. Evolution of split genes. It is supposed that split genes are the ancient condition, and bacteria lost their introns only after evolution of most of their proteins. Evidence for the ancient origin of introns has obtained by the examination of the gene that encodes the ubiquitous enzyme triosphosphate isomerase (TPI). The TPI is coded by a gene that contains 6 introns. In vertebrates, 5 of these are present at the same position as in maize. This shows that 5 introns were present in the gene before evolution of eukaryotes about 10 to the power 9 years ago. The TPI plays a key role in cell metabolism that catalyzes the interconversion of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, a central step in glycolysis and glycogenesis. By comparing this enzyme in various organisms, it appears that the TPI evolved before the of prokaryotes and eukaryotes from a common ancestor cell progenote. The unicellular organisms under a strong selection pressure minimized the superfluous genome in their cell, whereas there was no such pressure on multicellular organisms. That is why Aspergillus has 5 introns and Saccharomyces has none. Precise loss of introns would have occurred by deletion in prokaryotes. The TPI is thought to be evolved to its final 3-dimensional structure before eubacteria, archaebacteria, and eukaryotic lineage split off from progenote. G. Overlapping genes, genes within genes. In 1940s, Beadle and Tatum proposed one gene, one protein hypothesis, which explains that one gene encodes for one protein. However, if one gene consists of 1,500 base pairs, a protein of 500 amino acids in length would be synthesized. In addition, if the same sequence read in 2 different ways, 2 different amino acids would be synthesized by the same sequence base pairs. It means, the same DNA sequence can synthesize more than one proteins at different time. It was realized for the first time when the total number of proteins synthesized by exceeded from the coding potential of the phage genome. A similar phenomenon is found in the tumor virus SV40, where the total molecular weight of proteins, i.e. VPI, VP2 and VP3, synthesized by SV40 genes is much more than the size of the DNA molecule, 5,200 base pairs, i.e. 1,733 codons. From this observation, the concept of overlapping genes has emerged. For the first time, B.G. Barrell and co-workers in 1970 gave the evidence for the possibility of the above fact based on the overlapping genes found in bacteriophage OXY74. This virus contains an icosahedral capsid with a knob at each vertex enclosing a single-stranded circular DNA, Fig 2.8. F. Sanger and co-workers in 1977 mapped the whole nucleotide sequence of phage OXY74 and phage G4 DNA. The sequences of genes D, A and J, and B, to overlap in the whole sequence of OX174. The OX174 strand is made up of 5,386 nucleotides of known base sequences. If a single reading frame was used, about 1,795 amino acids would be encoded in the sequence, and with an average protein size of about 400 amino acids, only 4 to 5 proteins could be made. In contrast, Oxy74 makes 11 proteins containing a total of more than 2,300 amino acids. The sequence of gene A is now known to contain all of gene B. Gene B is translated in a different reading frame from gene A. Similarly, gene A is encoded within gene D. Another translational control mechanism expands the use of gene A. The 37K Dalton gene A asterisk protein is formed by reinitiating translation at an internal AUG codon within gene A message. The two translational proteins are synthesized by the same translational phase, but the functions of the two proteins differ. Protein K initiated near the end of gene A includes the base sequence of gene B, and terminates in gene C. For example, a reading frame of G AG TTA ACA. Nucleotides encodes the amino acids lysine, leucine reading frame of an threonine. However, after reading the frame one nucleotide earlier, the codes become GA, GTT, AAC, A, that encode glutamine, valine, and asparagine, respectively. It is obvious that by shifting the reading frame, i.e., overlapping the code, the same gene can encode two different proteins. Similarly, in the nucleotide sequence, TAATG, TA acts as termination codon of D gene, and ATG acts as the initiation codon of gene J. Here, the nucleotide A between A and T overlaps between the two codes. Therefore, the amino acid sequence of A asterisk is similar to a segment of protein A. In addition, overlapping genes have also been detected in animal virus SV40 and tryptophan mRNA of E. coli. H gene organization. The DNA molecules that make up the hereditary elements are called the genome. The functional region of the genome is called genes. In a complex genome, only a small part is functional, in that it is coded into a protein with the amino acid sequence determined by the DNA sequence. Another small part performed regulatory role by determining the time and extent of decoding in the of an organism. Protein coding DNA, along with associated regulatory sequences, makes its sense. A major part of genome is composed of highly repetitive sequences of unknown function, which were previously termed junk or selfish DNA. The DNA is a linear string or symbols. A, T, G and C proteins are synthesized by reading a code from DNA sequence, with a triplet of nucleotides, a codon correspondingly amino acid. Since 20 amino acids are the constituents of naturally occurring proteins, and there are 64 codons, the genetic code is the generath. The genetic code also includes a rule for initiation of protein synthesis, the start codon, and a rule to signal the end, the 3-stop or nonsense codons, figure 2.9. The prokaryotic genes have no misprints or interruption, while eukaryotic gene is split into several discrete segments called axons, which are interspersed with non-coding intermediate regions, i.e. the introns. Exon may be mixed and matched in various combinations to create new genes. Sometimes exon of one gene may be intron of another gene. The entire gene is transcribed into an RNA molecule from which introns are spliced out, resulting in mRNA. The mRNA is translated into corresponding polypeptide. There are also ancillary regions or the DNA which regulate and control the expression of proteins at specific time and under specific conditions. There are several ongoing projects to sequence the entire genome of a number of organisms. The complete genome map of some important organisms may come within a few years. For example, Drosophila melanogaster genome length equals 165 million BP, consisting of equals genes E. coli 4.7 mbp, 3000 genes Saccharomyces cerevisiae 12.50 mbp, 6400 genes Arabidopsis thaliana 1 mbp, 13100 genes Nematode Cyanohabditis elegans 1 mbp, 1500 genes Fugu rubrites 390 mbp, 8000 genes, and the human genome. Recently, entire genome maps of Haemophilus influenza L.83 mbp, 1727 genes, and MV coplasms genitalium 0.58 mbp, 482 genes have been sequenced. L. Gene expression.